Welcome everybody to part 2 of our API crash course. I'm Dennis Peel. I'm a software developer with the RCSB Protein Data Bank based out of our Rutgers University data site. And this is going to be our more hands-on tutorial, like one-on-one workshop for our APIs. So I assume that everybody here attended the first part of our crash course, which was held last week. And that was more of giving a demo on and an overview of our APIs. And now this part is going to be focused more on hands-on questions and letting you work through problems of your own while also getting help from tutors that are available. So just a quick review. Part 1, we covered an introduction to PDB data, how it's organized, and how the API schemas are constructed and where you can find information about our API schemas. We also provided an overview of the data API tools and applications that you can use it for, as well as an overview of our search API. And then we ended with a quick demonstration of a search API, data API pipeline, where you can combine both of the APIs in a very simple example case. Those recordings are now available on YouTube. So if you were to go to this link here, and you should have been emailed a link to, but in case not, this is the link here. It'll also be provided on our PDB 101 webpage under our training events. This will come soon. We're planning to also add this recording for this part to that website. So once we get this recording all uploaded, we'll release that website. So just keep an eye out for that on PDB 101. And we're also going to provide the slides and notes that came with the first part in the YouTube description, as well as on PDB 101. Shoot. All right, let me stop sharing. Sorry about that. It's not full screen anymore. There we go. All right. So today the agenda is shown here. We're going to start with some examples given by our tutors, Sebastian Bitrich and Joan Segura. And they're going to walk through some Python examples with you of using our search and data API. And these will be all provided on a Google Collab notebook, so you'll be able to follow along and test it out yourself. Once we go through that, then we're going to break out into smaller groups, and you'll be able to focus on your specific questions of interest. And then at the end, we'll all come back together and report out any interesting stories or questions that came up during the breakout groups. Some minimum requirements for this session, and just to set up expectations, is that first of all, you attended or at least reviewed the part 1 session, that you're familiar with Python and some of the basics of Python programming, that you have a Google account that you're logged into right now, because you're going to need to access the Google Collab notebooks. And we also hope that you have some example research questions or use cases that you want to investigate. And if you don't, that's okay, as long as you come with an eagerness to work together with another participant on their use case. So if you join a breakout room and somebody else is asking a question, hopefully that you can learn something from the questions that they're asking as well. And then, of course, we hope that you bring a passion for learning new and powerful API tools. So the breakout rooms that we're going to offer are going to be in these following categories. So depending on what question you want to address, you can join a particular breakout room to work through that question. So the first breakout room will be for general search questions. If you're just interested in general usage of our APIs, how to search for data, how to apply filters and criteria to your queries. And then also other technical questions, you're welcome to join this first breakout room. The second breakout room will focus on structure similarity search and structure motif search. So if you're interested in parts of a structure and finding structures that have similarities, you would join that room. If you're interested in studying protein complexes and interfaces, you can join the protein complex and interface group. And if there will be another breakout room for the annotations and positional features related to protein structures. And then the last breakout group will be for chemical search and ligand annotations. So some resources to keep in mind for this crash course, or this part of the crash course. We are also offering another Google Doc for this part, which is shown here, and it should also be provided in the comments section. So go ahead and open it up, and I just want to quickly demo this. So I'm just going to open it up here. And at the top, we have an agenda, and then where it'll link to the different parts of the document as you click on those links. And we also provide some resources for you to quickly reference. So feel free to click on any of those links for quick reference to information. And then if you have questions, feel free to ask them here. And then each section will have its own Q&A section. So try to find the appropriate one for the question that you're asking. And then we'll have a separate section for the independent and small group work. So if anything interesting or you want to take any notes, we encourage you to do so by adding them here under whatever section that you're in. And then we have a smaller section here for any closing remarks. So the links for the Google Collab notebooks that you'll be looking at and working through soon are provided here, and these are also linked to in the Google Doc. So again, this is a good way to quickly get to these links. All right, and so with that, I'd like to just start with acknowledging everybody from the RCSB PDB team who makes our resource possible. In particular, I want to highlight the tutors that are going to be helping out today. Those are Jose, Joan, Shenghua, Yana, Sebastian, Brinda, and myself. And I'd also like to acknowledge Shamara and Michael for all their help in organizing this meeting. And I'm done. Stop sharing. And I'm going to now hand it off to Sebastian to provide the first group hands tutorial. Oh, sorry. One last thing. I just want to let everybody know we are recording this first part of the section. So this first part will be made available later. Thank you. Take it away.
Thank you, Dennis. So, hello and welcome. So as you heard, for the second part of the crash course, we use a more interactive format. And to accomplish this, we set up Google Collab notebooks. And I just posted a link in the chat, so you can open it in your browser and follow along. And in case you've never seen Google Collab or Jupyter Notebook, I will give you like a short, like the idea is that you have basically this web page with additional information. And then most crucially, you also have what's called a cell, which can contain, in this case, Python code, and you can directly execute it in your browser. So it's a really convenient way to start scripting and to develop your code. And basically, if you see this document, you will have your own copy of it. And for every snippet of code, you can click this play button and it will execute this particular cell, and it will give you some response if it's programmed to do so. And what's happening in this example is that we use the requests library to make a request to the RCSB homepage, and we just output the status code. So here we see 200, which just means, yeah, the page is available and everything is working fine. And don't want to go in too much detail, but again, like the gist of it is you can use the system to follow along and run this code directly in your browser. As a participant, you can't make changes to this notebook, but you can create your own copy of it. At the very end, you can use the save functionality to basically persist whatever you developed, or if you maybe have some scaffold that you want to explore further down the road and keep working on some code that you developed, you can save your particular Collab notebook, and then you can also share that with other people. And to give you some starting point, we added some small examples. So first, we want to interact with the RCSB Search API. And you already heard a lot of information on this. So I'll just show you a simple query that looks at one attribute, the experimental method, and it ensures that it exactly matches X-ray. So basically, we're querying the whole archive for X-ray structures. And again, as you come across one of those cells, you want to make sure to execute it so that this query definition gets assigned to a variable. And then in the next step, we can use this variable to actually launch a request to the Search API. And to do that, we just specify the URL of the Search API, and how we interact with it in this particular case is that we provide a URL parameter called JSON. And to do that, you just, it's good enough to concatenate the base URL for the endpoint with a JSON representation for this query object that we defined up here. And then we can use the same package to launch a request to this URL with all the information that we provided, and we will get some response. And this response happens to be JSON, and we can then print it to the console. And if we execute this cell, you basically see the same thing as you would see in the query editor if you use that to interact with the Search API. So you can see we got 180,000 structures that were solved by X-ray diffraction, and you can see the first 10 identifiers.
And the respective score, which is the same.
It's either, we solved using X-ray diffraction or it isn't. So that's like a simple way to interact with Search API. And we also have this alternative way that we already showed to you. So there's a dedicated Python package called RCSB Search GPI. And if you want to use this, you need to first install it. And one big advantage of this tool is that it allows you to define queries.
You were breaking up a little on my end, Sebastian.
Yes, he's breaking up for me too.
Yeah.
Maybe I should let me stop video, but I hope it gets a bit better.
Okay.
Let me know if it's
help.
if you didn't hear anything. So basically, we install this package, and what it allows you to do is to define these query objects in a more convenient way, because there's still some boilerplate that you need to structure your query correctly to actually launch it. But this package allows you to define an attribute query, and you just need to know what attribute are you looking for, and then you even get some autocomplete where you can say, okay, maybe you're not exactly looking for a phrase, but you just want to have something that contains a particular phrase, depending on which attribute you're actually looking at. So it helps you define queries, or makes it a bit more easy to find correct query definition. And in the very end, nothing fancy happens here. So it's just another way to launch a query to the Search API, and then you get basically the same output. So here I configured it to give you the first 10 identifiers that match this criteria. So the response is pretty much the same as we saw before, and you also see the same number of structures matching this particular query. And the other API that we want to interact with is the RCSB Data API. And this one behaves a bit differently. But what you want to do to pull information from Data API is basically define a snippet like this, which defines which information do you want to retrieve for a set of entries. So in this particular case, we are looking for the experimental method. So it's basically similar to the attribute that we used in the Search API query. And we also define which entry IDs we are actually looking for. So in this case, we are just pulling information on one single entry. which was part of the results set above. So we would expect this to be also solved with X-ray if all goes well. And because interacting with like any GraphQL endpoint can be a bit tricky, we also use a dedicated client for that. So it's another package that we want to install. So just execute the cell and it will install the Python GraphQL client, which makes it easier to dispatch queries and also makes it easier to handle potential errors. And with that, we have everything that we need to launch this query. So we just create this client. We point it at the RCSB data API endpoint. And then we just pass in the query that we defined above and the set of structures that we're looking at. So again, just a single structure, and we can then, again, nicely print it to the console. So this really basic overview of how you can interact with Google Collab notebooks. And so now it's 20, maybe let's wait until 22, and you can follow along, execute those individual cells, and you can start developing your own code based on these like minimal examples that allow you to interact with both Search API and Data API. And one thing I didn't point out, you can easily make changes to those individual cells. So you could, for example, pull more information for one particular entry, or you could change the Search API query to look for something more elaborate that is more closely related to the use case that you actually want to address. So yeah, let's continue at 22 and let us know if you have any questions that came up at this point. So just basically make sure that you can execute code.
Just a reminder to everybody too, if you decide to modify anything in the Collab notebook, make sure you save it as a new copy on your own cloud. Because if you just exit out of it, you'll lose those changes. Thank you, Sebastian, for the great tutorial. Okay, it doesn't look like people are raising any questions now. Of course, if you do have questions on it later, we can raise them on the breakout rooms. So for sake of time, let's move forward. Joan, would you like to provide your walkthrough now?
Yes, of course. Thank you, Dennis. And thank you all the participants for attending to this webinar. Okay, so I will present a more practical example. In this example, we are going to see how we could compile a data set for setting up our machine learning method. In particular, we will focus on the potential problem of prediction of protein-protein binding sites. So in the top of the notebook, you will have important links to the Search API documentation, Data API, and to the query editors. Really useful tools when you are trying to set up your queries and checking what is the data dispatched by the services. So in the first cell, again, we will need the Python GraphQL client in order to request information to our GraphQL services. So let's first install this library since it's not native with the Python installation. Okay, so before that, a few observations. Please take this really careful since they are only partially true. So after this talk, forget them. When I am talking about PDB instances, you can think about PDB chain. When I will talk of a PDB entity, you can think on sequences. So if you want a deep and clear description on the PDB hierarchy, please go to rcsb.org, because we have a really nice and deep documentation on the different terms of the PDB hierarchy. So when I use the terms document, dictionary, hash, map, or record, what we are saying is encoding this information in keys and values. No, for example, a record for a person could be name and age. Also think that the values can be other documents, no? So for example, we can have one of the attributes of my document, date of birth, and will contain the year, month, and day. So when you see some attribute that is defined by a string concatenated with dots, what we are pointing is an attribute that the path is date of birth and year. So take this into account, because in this type of this way of encoding the attributes will appear on our search API. So let's first of all set up our URLs for the search API and the data API into a global variable, so we don't have to use the whole string. And let's go to our example. So in this case, what I'm going to explain is how we could set up a data set for prediction of protein-protein binding site. In particular, we will be interested in predicting binding site for heterodimeric complexes. I will explain a strategy how we could deal with redundancy. We don't want the same assembly or the same assembly composition to be present many times in our data set. Otherwise, we can just bias the method. Also, I will explain how we could split our data set into a training and independent testing set. We don't want to cheat. If the training set contains information about the testing set, we could be just having a performance much better than the methods is able to predict. I will explain how can we find the binding site for the assemblies. I am not sure if we will have time to reach to this part. However, you will have all the code to do that. So the general strategy will be, we will request assemblies to the search API, and we will enrich those assemblies with biological annotations coming from our data API. Okay, so our first query to the search API. So we said we are interested in heteromeric assemblies, in particular heterodimeric assemblies. And as we want to predict protein-protein binding site, I think it's fair to say, okay, we want the sequence of my proteins to have a certain length. So here I have chosen 30 amino acid length, but it's true that there is no definition when a linear peptide becomes a protein. But I think all of us will agree that the 4 amino acid length sequence is not a protein, can be qualified as a linear peptide. So, okay, let's inspect this search query. And for that, let's go to the query editor. Okay, so here we have. So these tools are really useful because they allow us to know what is going on. We can, before dealing with the data, we can check if the query is right, if it is not right, and we can inspect. We have a first idea of what type of objects are we getting. So in that case, our request contains 3 different conditions. We said we want heteromeric assemblies, they need to be dimeric, and the sequence length needs to be greater than 30. And we can see that in these three different attributes here, these three different nodes. So in that case, we saw Sebastian that he presented a single query. So in the query attribute, we didn't have a group of nodes, but a terminal node, straightforward. In that case, as we want to combine three different attributes, what we can do is define our query as a group and just use a Boolean operator. In that case, what we want is all conditions to be true. So now the question, I think, is how can we find those attributes here? So for that, we have the search documentation that it's really cool because it provides great details of all possible configurations. And we have this section here on the text attribute. So they are divided between the ligand, so small chemical attributes, and what are the protein structures attributes. So in this case here, you have a list of all searchable attributes. No? So let's see that, for example, this polymer composition. So is part, oh, sorry about that. Yeah, so it's one of the searchable attributes. And here you have a description of what is attribute, what type of information this attribute is encoding. You know, categories describing the polymer entity composition for the generated assembly. So in this column here, we can find what operators the attribute accepts. And what it's a bit difficult to decide is why here I am using this value heteromeric protein. So if this is defined as a string is a free text, so it could be anything. This is not true. So for technical reasons, we need to define that as a string, but this is a controlled vocabulary. And how can I know this value? So one of the things that you can do is go to the data API. And here I want to highlight all our services use the same schema definitions. So whether an attribute is defined in the search API or the data API, all of them are coming from the same schema. So it doesn't matter, both APIs will explain the same thing about our data structure. So in this case, in the data API, you have this really nice tool that is the GraphQL that we will use it later to play with queries. No, it's similar to the search query editor. And what is really cool is that you have here the documentations for all our queries, all our endpoints, and you can search by any of our fields, that any of the attributes of all the documents stored in our database. So in that case, we are interested in assembly information, polymer composition, and here you have a description of all possible values for that attribute. In that case, we were interested in heterodimeric proteins. Another thing that you could do is use the rcsb.org advanced search. And now one can be wondering how a UI can help me to decide my search query to the API. So first thing, what is really nice is that you can search all exposed attributes for search in the UI. So for example, if I am interested in the composition, okay, so I can easily find a field, and then it will provide automatically what are the allowed values. So I could just go here, search, and first of all, you will have the chance to see if the results what you are getting are really what you were expecting. So what is the connection with the search API? Because there is this nice function here that will lead you to the query editor where you could see the query. And maybe the query, it is not exactly what you want, but at least it's a starting point. So here, for example, it seems that the polymer composition is at entry level and not at assembly. But at least you have some ideas and you don't have to go straight forward to the attribute list and go one by one. You have some ideas of what you are looking for. Okay, so let's execute this query. And yeah, okay, so just remember two different things I want to highlight here. Return type, so we are interested in assemblies. So our search API can dispatch any level of our hierarchy. We can request for entry information, assembly, instances, or entities. And the second thing is, as we now integrate experimental and computational models, so you have to be careful, because for our case, we are interested in setting up a method for protein binding site prediction. And for that, we wouldn't like to be using data that is coming from other prediction methods. So we just set up that our content type must be experimental. So here, just a Python code, how to execute that query, similar, quite the same as Sebastian did, using the native request method that comes with the Python library. This allows us to do HTTP request, and the JSON is the library that we use to parse the response, not to convert these JSON responses into Python dictionaries. And the only thing that I am doing here is just parsing the total number of, storing the total number counts and the result sets, in particular, the identifiers for each of the items coming from the result set. I'm plotting here the first 10 assemblies that we received from the search API. Okay, let's go now to, I have my list of assemblies. Let's talk a bit, how can I use the data API to annotate information on these different objects. So let's first talk about simple requests. So a bit on the query structure, on the GraphQL query structure. So the first line is just to give a name to our query. It will also allow us to set up parameters. So I can define a static query, and I only have to change the parameter value in order to get new data. So I don't have to modify my query. So the second line of the query, it points to the entry point, to the query itself. So you can think about an entry point as a pointer to a particular collection in our database. In that case, our assembly collection, so the collection that contains the information for the PDB assemblies. One of the really useful things of this tool is that you can Alt-click or Command-click in macOS, and you will redirect to the documentation of the attribute or the query. So in that case, I am requesting assemblies, but what I want to highlight here is all our collections will always expose RCSB ID, so identifier for any document. And these RCSB assembly container identifiers that contains a more granular description of the document identifiers will be available for any entry point. So with this, what I mean is, if instead of requesting assemblies, I am, for example, requesting information for an entry, what I always will find is the RCSB ID and something that is RCSB name of the collection or relate something close to the name of the collection containers identifiers. And here I will have the entry ID. And this is the same for instances and entities. The other thing that I want to highlight is we know that PDB data is hierarchical. So the GraphQL API follows this hierarchy. So what I mean with that is if I am requesting for entries, okay, so for example, we know that entries encode different assemblies. No, an entry can have different assemblies. So the entry point assembly is accessible from the entry collection, so from the entry query. And here again, we see that we have always RCSB and the containers identifiers. So in this case, let's use another case where I have more assemblies. Okay, so what I see is that this entry here, 1XX, contains two different assemblies, no, the assembly 1 and the assembly 2. But okay, so what is an assembly made of? Chains. So inside assemblies, I have also access to the instances, no, polymer entity instances, to the chains. And again, RCSB ID is always going to be an attribute, and RCSB containers, in that case, RCSB polymer entity instance containers identifiers, is going always to be available here. No. So in that case, when I can generate for an entry the list of assemblies, and for each assembly, I can have the different instances that are found in the assembly. Okay, so this is what I am using in this request here. I am requesting assembly information. I am capturing the identifiers of the assembly, and I am also requesting the information for all the chains into the assembly, as we saw in the GraphQL tool. So when we execute that query, we don't do fancy here, just parse this data. We are storing the assembly as a list we are storing all the assemblies in a list where each assembly is encoded as the list of instances. So when we execute that query, so this is what we get. No, in this case, here we have the information for us first assembly. I have the assembly identifiers, the PDB entry ID, the entity, and the chain ID of the first chain of my assembly. Just I want to notice two things. So this is a list of lists. The result is a list of lists because I am getting a list of assemblies, and each assembly is being stored as a list of chains, no? And all of them are going to be of length 2. So all assemblies are going to have the same length because I have previously requested information to the search API of heterodimeric assemblies. Okay, so let's now add some entity level, sequence level information. So again, as we said, the GraphQL interface API follow the same hierarchy as the PDB defined hierarchy. So inside instances, I can access the polymer entity information. So if I come here, so one, again, so this is instance. I know a chain is a representation of a PDB entity. So if I search here for entity, I will have what is the relationship between instances and entities. Again, remember, RCSB ID is always an attribute of our documents, and we have the container identifiers attribute that it contains all the IDs for the documents. No, so in this case now, what I am adding, this might be now getting a bit messy, but for each of the instances, I am also having the entity that this instance represents. And of course, inside Polymer entity, we can have the length of the sequence. The question is how to find this attribute here, how I know that Polymer entity poly, RCSB sample sequence length is the field to find the sequence length. Again, remember, you have the documentation, this really nice tool exposed in our GraphQL documentation. So when you search by length, you have here entity poly RCSB sample length. No, so it's true. Maybe you have to search a little bit, but it's easily, at least makes easy to find the attributes that you are interested in search into getting the data. So what I am doing in this query is pretty simple. I am just now adding to the example before, so to the previous request, the sequence length information. So we can now execute that, and what we are getting now, so I am displaying the information for the server assembly. Remember, our assembly are encoded as a list of instances, and as are heterodimeric, I will have always two instances, so two elements in this list, and I have the information for the first chain and the information for the second chain of my assembly. And the question is, why do I have a length for here when I requested in my search API that I want the sequence length in greater than 30 amino acids? How is that possible? Yeah, this is one technical limitation that we have in our search engine. When we have a relationship that involves one-to-many, so for example, one assembly contains multiple chains, there is an issue here. We have to fix the behavior. We can choose whether this will behave as for all or for any. So the condition needs to be satisfied for all elements or for any of them. And in this case, once this is selected, it cannot be changed. Well, we can change it, but it cannot be changed dynamically. This needs to be changed in the process when we index our database. So in this case, the behavior is going to be for any. So if we want to filter that, we can do it on the consumer side, no, on the client. It's really pretty straightforward. What we can do is now just filtering. I only interested in those assemblies where the sequence length will be greater than 30. So this is not, this is just the code that, again, it's parsing the GraphQL API response. And here what we can see now is that the first assembly has a length, both chains in the assembly has a length greater than 4. No? So previously, the previous assembly has been filtered and removed for that. Okay, so as we include sequence length, we can also include Unipro information. And what is the motivation of including Unipro information? Well, if you want to build a non-redundant data set, one approach that you could use is that I don't want to any Unipro combination appear twice in my training set. No? So all assemblies will have a unique combination of Unipros. So we could easily just add Unipro data, so the Unipro accession to our chains. This is all also part of our Polymer entity collection, and the Unipro side is the attribute accessible from the Polymer entity container identifiers. So again, nothing special here, just parsing the response from the data API. And here is what we have. Okay, so I forgot to filter here the length. So I get again the length 4. And yeah, what I realized is that some of our chains might not be associated to any Unipro. And yeah, that's true. So if this is a length 4 protein, so maybe this is a synthetic peptide. So and it is not related to any Unipro. But not only that, so antibodies, or I can have a whole synthetic protein that won't be related with any Uniprot. So maybe adding Uniprot information is not the best strategy that I can use to create a non-redundant data set. Okay, so the only thing that I'm doing is again adding the filtering step, so we make sure that all our chains are greater than 30. Okay, so the previous assembly has been removed now, and in that case, the first assembly has both Uniprot. But this does not guarantee that all the chains in my assemblies will have Uniprot. So what can I do? Well, one of the things that will be available is the cluster IDs. And what are the cluster IDs? So every week, we cluster all the sequences in our database, no, from sequences coming from the experimental structures and from the computed models. So what we do is run a cluster for different threshold or sequence identity, and once these clusters are set up, what we do is identifiers. Then we give a different identifier, unique identifier for all the clusters, and we annotate the group ID into the sequences. And those group IDs are again accessible from the polymer entity query, no, or polymer entity entry point in the data API. So they are accessible through the RCSB polymer entity group membership. And what here I am just collecting is the similarity cutoff and the group ID. Now let's see how the results of this request look like. Again, filtering by 30, nothing fancy, just formatting a little bit the JSON response from the data API. And here is what I have. Now, this is the information for the first assembly. I have, again, all the identifiers, entry entity, chain ID, the length of the chain, the Uniprot ID, and here I have the group IDs. No? So, okay, so I have many group IDs. What is going on? So remember, we compute independently clusters for different sequence identity threshold. So for each threshold, we have a completely separated cluster. And this is what these different group IDs represent. They are always following the same format. So first, an integer identifier concatenated with the sequence similarity threshold. So what I can do now is, instead of using the Uniprot ID to create a non-redundant data set, what I can use is the group ID at 100% of similarity cutoff. Why? Because this will guarantee that all my assemblies will be different in terms of sequence identities. None of them will share exactly the same sequence composition. So now, again, nothing more than parsing the request, but now instead of storing all groups IDs, I am just focusing on the group ID that is similarity cutoff is defined at 100%. So yeah, so sorry about that. Here is the one I execute that query. Here is the response that I get. It's just now what I have had is the group ID at 100% of sequence similarity. Okay, so let's talk now a bit of annotations in our data warehouse. So similar to what we were describing before, that all collections have these RCSB containers identifiers, something similar happens with annotations. Of course, that not our objects will contain annotations, but all the collection that annotates data, so they have biological annotations describing some properties of the objects, will have a common pattern that will be RCSB, in that case, for example, polymer entity, polymer instance annotation. Let's see to the data API, to the GraphQL tool, some examples of that. So, okay, so here I am accessing the polymer entity instance collection, no? So the entry point, so chains. And in that case, we have the RCSB polymer instance annotation attributes. And what I get here, so okay, so in the case of our instances, what I get is protein domain classification. I have cath, I have SCOT, I have ECOT annotations. But what I was trying to explain before is that if instead of polymer entity instances, I am interested in polymer entities, and the attribute in that case is entity IDs, I don't have any more polymer instance annotations, but I am sure there is some kind of annotation here. It's called polymer entity annotation. And yeah, what I get here is, okay, so these are, again, protein domains, but instead of based on structural information, derived based on sequence information. Well, we have here a glycoprotein, so I'm not sure if this is a protein domain. Probably is related with glycosylation. But here we have a p-fine domains, no? So yeah, and the really good thing is that all annotations follows the same data structure: name, type, annotation ID, and probably there are other fields there. So what I can do.
Sorry, Joan, we're approaching the end of the session, but feel free to.
Okay, so let me
summarize.
finish at least as part of annotation, because and then let's people, if they are interested in continue playing with the example. So yeah, so what I am doing here is adding to my assembly data information on polymer instances annotations. Okay, so let's just execute that. And let's see what we get. Again, nothing fancy in the code, is just parsing and just restructuring a bit the data. So now, for example, the information on the first assembly, what I have done is adding all these domain classification information. And what I am doing that, well, because what I could do is, okay, instead of storing everything, let's add only information about cath domains. And what this will provide me is a way of splitting my data set between training and testing. So one of the strategies to avoid that the training contains information on testing, you can say, okay, I can define that two assemblies are independent if they don't share the same cath composition. So what I could do is just splitting my data set into a training test and testing set where none of these assemblies will share between training and testing, will share the same cath composition. Okay, so next pieces of codes follow more or less the same patterns that we have been seeing and are related on collecting interface annotation that is going to be defined as positional features. So, but I will stop here. The information is there, so please feel free to ask any questions and play yourself a little bit with the Collab. Okay, I'm going to stop sharing, Dennis.
Thank you, John. That was a exhaustive tutorial and very thorough. We hope that everybody finds value in running through that on their own. And specifically, you were ending on the protein complexes and interfaces section. So that's actually a convenient place to leave off because we're about to break up into breakout rooms. And one of those breakout rooms is for complexes and interfaces. And Joanne will be hosting that room. So if you're interested in that topic, feel free to join that room. So yeah, thank you to both Joanne and Sebastian for their great walkthroughs.